3 to 5. And yeah. All right. So today we are going to have the first question.
Sir, 3 to
Yeah.
5 or 2 to 4? Like last class, you said 2 to 4.
2 to 4, 2 to 4. Yeah. Sorry. 2 to 4.
Yeah, okay.
Yeah. Um, so today I'm going to introduce you the first problem, which is about finding the origin of replication in bacterial genomes. It's somewhat of an algorithmic warm up. We'll be going slowly into the problem, and you will see that rapidly the problem will become more and more complicated. And so, yeah, I hope it excites you enough so that you don't sleep and follow what I'm saying. Please stop and ask me questions whenever you don't follow something. Okay, so this is the outline of this module. First, I'll introduce you to DNA replication itself, a few experiments which tell us some information about how DNA replication works. Then I'm going to talk about hidden messages in the replication origin. So basically, how does the cell recognize where to replicate, where to begin replication? Then this is going to uh, segue into an algorithmic problem, which is hunting for frequent words, typically hunting for frequent words in a window of DNA, in a stretch of DNA nucleotides around around the origin of replication. We are going to come up with a very simple, what's let's say a greedy way or the most simple way of solving this problem. And then in the next next set of slides, I'll be talking about a faster way of looking at uh, counting the frequency of words. And here we'll be dealing with a lot of pseudo algorithms. So pseudo algorithms are basically way of writing a computer program in a more human readable format. And once you have a pseudo algorithm with you, then you can interpret or execute that problem in a language of your choice. You can choose C, you can choose Python, whatever is comfortable for you. Then we're going to go into some statistics of this problem as to when do we call something as surprising, or when do we call something as more frequent than expected. And then I'm going to take you basically more into the biological implementation of this set of algorithms that we would have developed, specifically looking into asymmetry around origin of replication. So these things will become more clearer as the lectures go by. Again, the lectures by the end of this lecture, I'm going to put it on Moodle. So if you want to revise, if you want to see something, if I want to do some self-study, you can do so by following the lectures. Okay, so the most important thing in the context of DNA replication is what James Watson and Francis Crick talked about in their 1953 paper when they described the structure of DNA. And they said that it is not escaped our notice that specific base pairings, the fact that there is base pairing that we have postulated, immediately suggests a way of copying genetic material. So basically because you have base pairing, you have two DNA strands. One DNA strand can be a template to copy, to make an exact copy of another DNA strand, right? So one can easily become two can easily become four. So this is what the mechanism looks like. You have a double strand. This double strand can be unwound. And then each of these double strands, because an A is always going to pair with a T, you can essentially copy and you can have two double strands. So this is the basics of DNA replication. Now, there was a very interesting experiment that was done. But before I talk about the experiment, there were some hypotheses as to how these copying mechanism, how this copying was happening. So there are three proposed mechanisms. One proposed mechanism is that the copying is semi-conservative. That basically means that you have, whenever you have the original parental DNA, but after one round of replication, the two double-stranded DNAs that will be made are going to contain one copy each of the original chromosome or original DNA molecule. Then the other mechanism was that DNA replication is conservative, which essentially would mean that the entire double-stranded template is going to be used for copying another new, synthesizing new strand of DNA. So the new strand of DNA is shown here in black, and the original parental DNA is shown in orange. Or the other hypothesis or other model suggested for DNA replication was that DNA replication would be dispersive. That essentially means that randomly DNA replication is going to start along the parental DNA, and as a result of it, you'll get a mixture of new and old DNA molecules in the two replicated fragments. So can somebody tell me quickly what was the hypothesis that Watson and Crick put forth? Which one of these three?
So I think it was the semi-conservative one.
It was the semi-conservative one because they said that the one DNA molecule, single-stranded DNA molecule, can act as a template to synthesize the double-stranded DNA. So they were more going towards semi-conservative and maybe even a little bit of dispersive, because you can say that every time, if this is happening randomly, so each random part can either on this strand or the other strand can make a copy of the other one. Okay. So there was a very neat experiment that was done to check this. This experiment is called the Meselson-Stahl experiment, where they took one isotope of nitrogen, nitrogen 14, which is lighter than the most abundant form, nitrogen 15, and they introduced this isotope, basically bases that are nucleotides which are labeled with this isotope during the process of replication. Okay, so what Masons and Stahl do was they grew E. coli for many rounds of replication in your normal medium, which N15. So basically all the bacteria now gain the weight of the DNA is proportional to having N15 in the, or the heavier isotope in their DNA, and then they're transferred E. coli with heavy DNA with in the N14 medium. So now whatever new DNA is going to be made, it's going to have this lighter nitrogen in it. So now with this, they were able to try to sort of decipher what is the form of replication that is happening. So the key point here was that the daughter DNA will be lighter, right? All right. So now after one round of replication, if you have semi-conservative replication, right? What so what basically Melson and Stahl did, Messelson and Stahl did that this after one round of replication, they spun the DNA in a centrifuge. Okay. Um, can you tell me why would they have done this? What does spinning in a centrifuge achieve?
Differential separation on the basis of weight.
Basis of weight, right? So now if you had semi-conservative replication or conservative or dispersive, how is the separation going to look like? So after one round, in the case of semi-conservative replication, how many, let us say, the lighter DNA is going to form a separate band, right, than the heavier DNA. How many bands are you going to see in the semi-conservative replication?
So we'll only see
There is
one band.
single one.
Single
Yeah.
band, right? And would this band be lying above the parental DNA or below the parental DNA? So let us say you have both the cells. You have the heavy cells and you have the cells that are obtained after one round of replication, and you mix the DNA from both of them. So relatively, where would it lie?
Below the
In
head
between. So
DNA.
below the heavy one, but above the lighter one.
Okay, right. So basically by doing this, they were able to find out that, okay, if it would be conservative, then you would be having just the parental DNA band, and you are going to have another lighter band, which was completely be made up of N14, right? But this is something that they were not finding. So they found that, okay, there was two bands. One band was, there was just a single band that was your original, that was basically half of basically containing N15 and N14. So there was a single band. So what if there would be dispersive replication? What would happen? So after two rounds of replication, what would happen?
So again, we'll have a singular band in the
Are you sure you
process.
can have a singular band?
Oh, yes, sir.
So after 2 rounds of replication in semi-conservative, you're going to have one set of DNA which is totally new and one set of DNA which is shared, right? So you're going to have two bands now, one completely lighter band and one which is slightly heavier, right? As compared to the lighter one. And
Yes,
if
sir.
you would
This
also
was
add the parental DNA, so they will have three bands. But if it is dispersive, then in the case of dispersive, you are going to have
there is singular band rate because
a
like
singular band, right? Which is
it would
going to
be
be, yeah,
randomly distributed.
exactly. So by actually looking at 2 rounds of replication, now they were able to um divide DNA on only two densities and not just a singular density, which would indicate a dispersive replication. All right. So by doing this experiment in 1958, they were able to basically find out how DNA replicates, and it does so in a semi-conservative fashion, whereby you always have a single template, the entire single template playing the role of being the template for the next like single strand being the template for the next replication round. Okay. When you talk about this as a biologist, what you are thinking about is essentially a wound up DNA molecule which gets copied and it gets redistributed in two cells, right? So this is what a biologist sees. Now, as a computer scientist or as a bioinformatician, uh, what would you be seeing? You would essentially be seeing a string, which is basically a DNA string, right? Some complicated biological process happens, and because of that, this DNA string now has a copy of itself, right? There are two copies of these DNA strings. So now, as a biologist and as a computer scientist, we have to come together and do some bioinformatics, and we have to solve the question. The question being origin of replication. How does the cell know where to start replication? So the location where the cell begins replication is called the origin of replication, or in short, ORI. And biologically speaking, it has been seen out of repeated biological observations that the out of all the divisions that happen in the case of a simple cell like a bacterial cell shown here with a circular chromosome, a singular circular chromosome, the replication always begins at the same location. So this is an observation that has been seen by biologists over and over again. And this location is called the origin of replication. The question is, can we actually find the origin of replication, right? So if you were to ask a computer scientist now, you would have to formulate the problem, right? The problem of finding the origin of replication would be there you have an input, which is a DNA string, which is nothing but the genome of the organism for which you're trying to find the origin of replication. And the output is the location of the ORI, or the origin of replication in the genome. All right. So now this essentially comes down to finding a hidden message, right? Or finding this hidden location of the origin of replication uh, in a given string. So is it a computational problem? Do you think that this is a computational problem? Is a well-defined computational problem?
Yes, sir. In case we know the sequence of origin of replication.
Yes, but we don't know the sequence of origin of replication, right?
Yet we don't know, like uh, uh, we have learned like in molecular biology that it should be 80 reach, right?
Okay, but is that enough? That's not
No.
enough, right? So we have to come up with a better way of defining the problem. What if you ask a biologist in the lab to find out where the origin of replication? So I've already answered the question, but do you understand this approach?
Yes, sir. Like randomly removing sequences from the uh, DNA and then seeing like in which case it can't replicate, and then we'll know we have taken out the ORIC.
Origin
Or
of replication, right? So the computer scientist has the problem the way to solve it was, is there something hidden inside the molecule, inside the DNA string that has been given? And a biologist would be doing basically a different problem. Like if I chop out different parts, then if the right part is chopped out, then I have actually hit the origin of replication. So now, as when we have to come together as with the biological information and with using computer science, you need to have define the problem in a little bit more convincing manner, in a more formal way, where you can exactly get to the problem. So now let us look at the origin of replication of Vibrio coli, which is a bacteria, right? Typically, an origin of replication is about 500 nucleotides long, right? So now let's try to understand, is there some hidden message, or is there some information that we can pattern that we can discover in this Vibrio coli? Can somebody see and tell me if they find some pattern? No? If it's a no, then you can say.
So I can't find.
Right? So, but if you look closely, yeah.
So the sequence is highly A and T rich, and there are lots of repetition of T's.
There's lots of repetition of T's. Okay, good. All right.
And the sequence is also highly rich in A and T.
You can say that by just looking.
Yes, sir. Like
Okay.
so
So definitely there is a lot of T's, but also our eyes are sort of looking at more vertical lines than every C, G, and A are more circular circles like curves. So probably T's are also standing out a little bit more. But good, that's good. But if you look at specific areas of this, I mean, there must be a lot of locations inside the cell, right? Which might have a lot of T's and A's and T's. There has to be something specific, right? That tells the cell to start the replication here. So when we go deeper into this problem, we can essentially say that there must be some input text that the cell is looking at, right? There must be a hidden message in the form of a text in the input sequence. So in this sequence, there must be some hidden word or some hidden information that the cell is looking at. Okay. So this is kind of formalizing. So now the problem that you have sort of defined is computationally finding hidden text inside a problem, inside a given like a hidden text within a input string, which is going to be your genome. All right. So now again, we have two scientific problems. First is, given a bacterial genome, which is approximately 3 million base pairs, where in this genome do you have the ori? Okay. The second problem is, given you have an ori which is approximately 500 base pairs, what is the hidden message which says that the replication should start here? Okay. Are these two problems clear to everybody?
Yes, sir.
Yes, sir.
Yes, sir.
Okay. So now let's try to define the first problem formally, finding hidden messages in the origin of replication. Let's solve the second problem. Again, you have an input which is a string, right? Represents the ori. The output is the hidden message in the text. Okay. So the notion of hidden messages is not defined. So Ratnesh, you were saying that if you have I hope it was Ratnesh, right? Or Dev, who said that if you have a text, if you know the hidden message, then we can find it, right? Then it's not a problem, right?
Yes, sir.
Right. So, but what if I mean, we have to define. So is the hidden message, is it always the same message, or it could be a different message, right? So it in fact is not always the same message, as we will find out. So we know that DNA replication is mediated by a protein called DNA, and this protein DNA is typically binding to short stretches of nucleotides called the DNA box. And this DNA box essentially is like a message to the protein to say that, okay, come and bind here. Now, whenever the DNA protein comes and binds to a location, that essentially is where the replication starts. So this is basically DNA box bind to short segments inside the ori. So this is biological information that we know. So now Would it make sense to have multiple DNA A boxes or just to have one DNA A box? What do you guys think? What does
There
everybody
can
think?
be multiple DNA boxes.
So
I
it
mean,
should be multiple.
Why?
like uh, instead, like there can be multiple cycles of replication going on at the same time. I think we've seen that. There was an example about E. coli about how the doubling time for its entire genome is, I guess, 20 minutes, but doubling time for E. coli is only 10 minutes or something of this sort. So it's like there's multiple cycles of replication going on at the same time.
Okay. Something a little bit more easier. I would like to see that paper if you have. I hope it's not a CNN article.
No, sir, it
That
was an
paper is
example
not
by
published.
Professor Preeti.
Okay.
She
Just send me some paper, exact paper about that. Is there some other reasoning? Why would you think that there would be more DNA boxes than just one?
So I would say just like to increase the frequency of replication or like the efficiency probably.
Okay. To increase the
Like
efficiency.
if
So
there will be just one DNA box, then the probability that DNA box will encounter and find that origin of replication which is present just at one place in a bacterial genome would be very less, and it won't be like it won't be like always possible that it is finding that origin of replication and
okay. Okay.
starting replication.
So
So
the
that's why
more
it should be
boxes, so the more
more in
boxes are
amount.
there, the more you will be more efficiently you will be able to attract the DNA A protein itself to bind. There could be also an evolutionary argument that if you have multiple sequences, then just by chance, if one of the sequence gets mutated, right, then you have the other sequences for your rescue. So these are the couple of reasons that we would be having. So this is actually does this is what happens. So multiple DNA boxes give higher chance of binding and give higher fitness to the organisms which have more number of DNA boxes. All right, and this sort of goes to sort of confirms the only theory in biology, which is nothing in biology makes sense in the light of. Can somebody tell me? Can somebody fill in the
Evolution.
blank here?
Evolution.
Evolution, right? So whenever we talk about fitness or fitness essentially means that if you compare two organisms, one organism had a single DNA box, right, versus another organism had multiple DNA boxes. The organism which will have multiple DNA box is going to replicate faster. And as a result, if you were to start with two organisms, let's say organism 1 with one box and organism N with N boxes, soon, because the organism N is replicating faster, because the protein DNA is binding to organism like the DNA box much faster because higher probability, right? More boxes. So it's going to overcome, it's going to have more populations of the organism N, right? So this is actually what's happening in evolution as well. And over the course of evolution, the organisms have accumulated more number of DNA boxes so that the replication is more efficient. All right. So evolution is the answer here. So now the question can be formalized from finding a hidden message to the nature of those hidden messages. And I'm going to talk to you about the nature of those hidden messages by looking at the same Vibrio coli 500 base pair origin of replication region for looking at words which have surprisingly high frequency. So basically, whenever you have a long string, like this is an input string, any part of this string which is smaller in length than the original string is called a substring. All right. So we are essentially looking for surprisingly frequent substrings. So one substring which is surprisingly frequent was already pointed out, and that is TT, right? TT is quite frequent. But do you know any DNA binding protein which is only binding to two nucleotides specifically?
No, so that would be pretty inefficient.
H1, H2, histone proteins like
So there were two questions. So Vishantam, what were you saying?
So I would say like that would be like very like chaotic.
It doesn't
If
make
there
sense,
is a
right? Because
binding
just by
protein.
chance,
Yeah.
you would find, like, if you were to have any two base pairs, the probability of finding TT in those any two base pairs, like, is going to be 1 by 16, right? So that's quite a lot of, like, if you have a 3 million base pairs along bacterial sequence, the probability of finding two T's simultaneously is going to be very high. So if the protein is going to bind everywhere, right? So this is, we are not looking for very small substrings, but we are looking for slightly longer substrings. So those longer substrings, when we actually look at um, the longer substrings, we will start to like some pictures will start to emerge. So now coming back to the problem, we are essentially redefining the problem of finding the DNA A, like where in the origin of replication does replication begin, into a more formalized computer science problem where we get as an input a string pattern, right? And a longer text, which is basically your input genome, right? The string pattern could be the DNA A box. This DNA A box could be known, it could be unknown. We're going to see that. The output is the number of times that particular pattern appears in the text. All right. So can somebody tell me how many times does ATA? So for example, if this sequence given below is the input text and ATA is the pattern we are looking for, can you tell me how many times does ATA come here in this pattern, in this input genome, small
So
string?
3.
3 times?
Yes.
Right. So it can be 2 or 3, depending upon if you were to take overlaps or not. All right. So if you count overlaps, it's 3. If you don't count overlaps, and then it's 2. All right. So now the key point is that when we start to think about a string, we can start to think about a string in order to solve this problem as an array of symbols. All right. So whenever we are storing arrays, if you have, I think most of you have done CUL 101, right?
Yes.
So you were taught in Python or C?
The Python.
So Python.
Some of us were taught Java.
Python and SML.
Right. So in most of the languages, the arrays are zero index. That means the first element of the array is stored at the 0th location. Right. Now, if you have a text that I was showing you earlier, this can be thought of as an array of letters, right? Array of symbols, where specific location, like each symbol is corresponding to a specific element in the array, right? So now if I were to represent this specific three uh, 3-mer or 3-letter substring TCC, I can actually represent it in the notation of text 7, 10. Why do you think I am representing it in 7, 10 and not 7, 9?
So upper bound isn't included.
Upper bound is not included, but it also helps us to inherently calculate the length of the substring quickly, right? By just subtracting the last letter and the first letter. But you can do that otherwise as well. But it's just a notation, right? So it doesn't matter. So now, if we were to look at CGA, how would you refer to the substring in this array?
So text 0 to 3.
03.
03, right? All right. What about this one?
Text 36.
36.
Right. So now what did you notice? If you want to basically calculate the length, you can just do 6 minus 3 and you get the length, right? You can also have another notation. You can have it as 3, 5 and you can do 5 minus 3 plus 1, right? Doesn't matter. So this just depends upon the notation that you the rules that you are having in order to define your substring, right? So now that we have defined the substring, how do you think we can count, like if you were to solve this problem of how many times does ATA occur in this particular string, how do we solve this problem? Now let's try to figure it out. So the most simple way of doing it would be to use the same notation, right? So this is your original array and this small notation becomes a subarray, right? It's a contiguous collection of values in an array is called a subarray, right? So this TAT becomes a some small subarray and you have to basically count the number of times you encounter a unique subarray. So for example, this is your original string. Now you begin at location 0 and you extract these subarrays or substrings, right? And whenever it hits the one that you want to that you have as an input, right? So basically the input here was 80A, count the number of times you have 80A in your original string. You would say you would increment the count, right? So the count was 1, then the count was 2, the count was 3, right? And you are ending. So how many substrings of length k? This basically comes to the problem of how many substrings of length k are there in a string of length n. So here k is 3 and n is 14. So how many substrings are there? Different substrings?
12.
12, right? But can you tell me the quickly the notation? For any string of length n and a substring of length k, how many substrings would you be having?
So n minus k plus
N
1.
minus k plus.
Yeah, this is cool. This is clear for everybody. N minus k plus 1. Okay, good.
Yes, sir.
So now let's try to write a code to count the pattern of the number of times you have a specific pattern occurring. So your inputs is the pattern and the text. You begin with count equal to 0. K is equal to the length of the pattern, right? N is the length of the text. So for every integer I, starting from 0 to n minus k, starting from 0 to n minus k, right? So at 11th position, you ask if the text, the sub array from I to I plus k equal to the pattern. If it's so, you increment the count, right? Length typically is a built-in function in most languages, which tells you the length of a array, length of a string, length of whatever uh, data structure you're using to store information, right? So I hope this simple thing is clear to everybody,
Yes, sir.
right? So if I were to make changes in this, and if I were to ask you to fix or tell me what problem is there in this particular algorithm, you will be able to do that. So those are the kind of questions that I'm going to ask, right?
Yes, sir.
All right, good. All right, so now let's go to the frequent words problem. So you are given a string of length k and a k-mer pattern. So this is basically a string of length k is a k-mer, right? A k-mer pattern is a most frequent k-mer in a string if no other k-mer is more frequent than that pattern, right? So for every specific k-mer that you extracted within a string of length n, if that's the most frequent k-mer, then that's the most frequent k-mer, right? So that's the problem. So now the frequent word problem can be defined as a string of text k, and you are given an integer k, which represents the length of the substring. The output should be all the most frequent k-mers in that text. So what are the most frequent k-mers? Depending upon the k in that text. So is this problem clearly stated? I hope this you would agree that this is a more clearly
Yes, sir.
stated problem, right? Than the
Yes, sir.
hidden messages problem. Everybody agrees?
Yes, sir.
Good.
Yes.
All right. So now let's go back to the origin of replication. Input is a string text and an integer k, and we have to find out the most frequent k-mers in a text, right? So let us say, as an example, let's take this text k is equal to 3, right? Then the most frequent k-mer words would be ACG and TTT. If you were to just quickly look into it, there is one T, and then there is, if there are overlaps, there are two TTTs, right? Similarly, there's AGC, is AGC here and AGC here, right? So both of them are occurring 3 times. So let's try to solve this problem. So we create a array of length text minus k plus 1, because these are the number of sub arrays you'll be extracting. For each of these i's, you set count I equal to the number of times that specific text has appeared in the text, and then at the end you would go back to the count matrix, this array, and you will basically say what's the maximum value of this count I. All right, so how does it look like? So you have a text this, and you have k equal to 3. You go to the location first, And at this location, you have the first k-mer that you extract as ACG. And then after extracting this ACG itself, you go along you extract all the other 3 mers and you compare how many times you found an ACG. You found it 3 times. So you write the number 3 here. Then you go to the next location, you have CGT. Then you go from this location all the way down and you ask how many times, not from this location, from the beginning of the text all the way down, and you say, okay, you have found it twice. Similarly, you keep on filling up this count matrix, right? And now you would say, okay, at position 0, position 3, position 7, position 10, 11, and 14, we have those k-mers which are most frequent, right? Because these have the maximum count value. All right. So
Yes,
how
sir.
does the hello? Is there a question?
No, sir.
I'm just solving the problem in a very simple manner, and now we'll start to, like, once the problem is solved in the most simplest manner, then we'll start to optimize this problem. Okay. So I hope everybody follows this.
Yes, sir.
Yeah. Okay. So now how does this pseudo code look like? We'll call this so this algorithm find frequent words, where the input is going to be text and the length k of the k-mer that you're trying to find with most frequent, with the k-mer for which the maximum frequency you're trying to find. So you'll have you have an array of strings with length 0, right? You call this fric patterns. N is the length of the text. Count is the array, the same integer, the same array that I showed. Now for every integer between I from 0 to n minus k, you're going to extract the pattern, right? And now you're going to count the pattern. So this is pattern count is the function that we have already written before, right? Which takes input a pattern and a text, right? So the count I is going to be pattern count. And now after you have done this for every substring that you extracted in the word, you're just going to say, give me the max value of the array count. And now for every integer between 0 to n minus k, if your count is maximum count, then extract the pattern at that particular integer and append it to this list of most frequent patterns. All right. Then at the end, you are going to obviously find lot of these patterns repeated. So for example, you had ACG at 3 locations, right? So the ACG pattern is going to be repeated. So you have to remove the duplicates within these patterns that you have found, and then you have to you have to return to the user what were the patterns that were more frequent. Is this clear to everyone? And obviously, if it's clear to
Yes,
everyone,
sir.
you can probably see how you can improve this algorithm, right? There are many steps that you can remove, but you can remove, reduce, and improve. Yes. So
Yes,
this
sir.
is very inefficient. Let's make it better. So a faster frequent finding approach. So now whenever you're storing lists, right, or arrays, you typically have this kind of a arrangement where you have your index, so 0 to whatever length of your what your array is, right? And at each of these indices, you have specific values stored. So for example, this is a string array, and you have at specific locations in the array, you have specific letters stored. This is a numerical array. This is an array of k-mers, right? So at location 0, you have a ACG. At location 6, you have CAT, right? So you can basically have different kinds of arrays themselves. Now, instead of using integers as indices like we do here, so we use 0, 1, 2, 3, 4 to 8, right? These are integers as indices. Can we actually use patterns as indices, right? So this would specifically be very interesting and useful in the case of our frequent pattern finding algorithm. So what do I mean by that? So instead of saying at location 0, I am storing an H, I am going to basically, instead of having a 0, I'm going to have AA as the index itself. So this is something similar to making an index inside your a book, right? So if you have a book At the end of the book, you will have an index, which will be lexicographically sorted. And each of these words that you are having in your index, it will tell you in what pages you had that word, right? I hope everybody has seen an index in a book. I know it's kind of,
Yes, sir.
yeah. All right, great. So now this is essentially this kind of index making is called making a dictionary or making a map. And this AA, AC, right? These non-integer indices are essentially called keys, and the value you stored at these indices are called values. All right. So whenever you make a map or a dictionary, you have certain keys, and at each one of those keys, you have certain values stored. So in this example, I am showing you keys as strings and values as counts, but this can easily be changed. You can have strings stored here, you can have counts stored here, you can even have mathematical formula stored here. So let's make it very easy. Let's have keys as strings and counts as number of times those strings are appearing inside a text. So basically, whenever we try to look at a variable here, so in order to refer to this a variable in this map, you're going to essentially say AA, comma 17 is a variable in this map, right? So instead of talking about the text 0, 3, you are going to talk about AA, 17 as a variable inside this map or a dictionary. All right. So for example, in order to access any key in this dictionary, you are essentially going to say, let us say this map is called frequency, right? So if you want to access the GT, like the frequency of GT, all you have to do is you have to say frequency GT, and it's going to return the value 30, which is stored in the map here. I hope this is clear to everybody. So this is basically called creating a dictionary. This is something which is this is something which is very basic in doing any kind of a bioinformatic application, because you're going to be creating dictionaries based on indices, letter-based indices for most algorithms that we'll be dealing with in bioinformatics. Clear?
Yes, sir.
Okay.
Yes, sir.
All right. So note that in this particular dictionary, not every tumor is here, right? So how many tumors can you expect in a dictionary which is fully populated?
16.
16. Good.
Yes, sir.
All right. So now let's create a better pseudocode for finding frequent words. So again, we'll have a text and a K. We have an empty pattern array for the patterns. And we have another addition where we actually have a frequency map. All right. So this is an empty map which contains all the K-mers inside your text. Again, you take the length as the length of the text. Now for every integer between 0 to n minus k, right, you extract the text I into I plus k, right? That's your pattern. And now what you do, you initialize your map if this pattern has not been seen before. So you say, okay, this I have seen this pattern. So for that particular k-mer, the value becomes 1, right? If that pattern already exists in your dictionary, then what you do is you increment the number of times you have seen that pattern, because the first time you see that pattern, that means the value is 1, and every subsequent time you increment the value. That would mean that you are seeing this pattern again and again. So every time you see this pattern, you increment the value. All right. So now instead of going through and having that pattern matching algorithm, right, pattern matching function, you basically reduce that time by actually creating a different kind of data structure. So a dictionary. So again, you do the same thing. Max count would now ask you what is the key in which you would have the maximum value, right, in your dictionary. And now for all the strings in this frequency map that you have created, if the specific pattern is equal to the maximum count, right? So for each of these strings, it's the maximum count, then you essentially just return it and you append it to the patterns that you want to find, right? So by doing this, we have reduced two redundancies. One is doing the counting itself and the remove duplicate step that we had to do. right? So now basically what you have done is you have had a simple implementation of the problem. The most basic implementation, you saw that there was some issues with that implementation, which was taking time, which was slow, and you introduce a new data structure here. So the data structure being dictionary, right? So just like that, we will see in the duration of the course, that very often, in order to speed up or better solve these bioinformatic problems, we are introducing specific data structures that are relevant, and this dramatically speed up and make your life much easier when you are trying to solve this problem. All right. So I hope everybody was okay with this. So beyond this, we can even shorten this particular pseudocode by implementing a subroutine. A subroutine is nothing but a smaller function, something that you can group together. So the whole concept of making this frequency map, you can shorten it, and you can call it making a frequency map. So given a text, any text and a string k, this particular subroutine or a function is going to return you a frequency map, right, of every k-mer inside that text, right? So now you have two functions which give you a frequency map, and a function which based on this frequency map can calculate the maximum value, and then you can quickly find the frequency of words. Now, why am I doing this? Specifically, if you were to go and look at the origin of replication in Vibrio coli, you see that if you were to have a k-mer as value 3, you have the k-mer with maximum count 25 is TGA. If you have k-mer 4, maximum count 12 is ATGA, right? If you keep on increasing the k-mer length, specifically, you find that you have at k-mer length of 9, you find that you have 3 different k-mers of length 9, 4 different k-mers of length 9, which have actually count 3, right? So this is sort of surprising. So the most frequent 9 mers in the origin of replication, all of them occur 3 times. Is this surprising? Would you say that this is surprising?
So I mean, large sequences are used by proteins to bind to specific sequences. So I guess, yeah, these would be
These would be surprising.
these sort
If you
of.
look at these sequences, do you see something interesting coming up?
So all of them have GATC, or like most of them
Yeah,
have the
all the 4.
CTT, GATC.
All right, so let's look deeper into it. Obviously, there is something interesting here. So DNA has reverse complementarity, right? So the DNA, right? If you have AGTC on this strand, then complementarity to it would be GACT, right? So now if you actually look at these sequences, ATG, ATC, AAG, and specifically this sequence, ACT, AGG, right? This particular sequence, these two sequences are actually reverse complements of each other,
Yes, sir.
right?
Yes, sir.
So now, again, let's solve a smaller problem. How to reverse complement a sequence? You have a DNA string as an input, and the output should be the reverse complement of that text, something that you guys have to do very often in this course. So let's write a short possible pseudocode for it. Pseudocode is you have a text, you reverse that text, and you complement that text, right? Reversing a text is easy. Complementing a text is easy. For every A, you replace it with a T. With every G, you replace it with a C. With every T, you replace with the A, C with a G, right? So I leave you to write the code for this and expect a problem on this in one of your evaluation components. Okay, so now essentially this code is there, so you can essentially tell whether what's a reverse complement. So now you can easily split your work into 2 cases. So now what we see is that specifically in this 500 base pair region, you have 3 9mers, right? You have 6 9mers in fact, and these 6 9mers represent 3 9mers, which are reverse complements of each other, right? Now it would make sense that proteins like DNA A, they bind to both the strands of DNA. Does it make sense? Biologically?
Yes, sir. In case of replication,
Right?
it can.
Because DNA replication has to begin on both the strands, right? This is something that we know. You have the replication bubble forming. So this protein needs to bind on both the strands. So right, for given this information, this totally makes sense. But what does not make sense statistically is to find these 9-mer messages so frequently in this small region of origin of replication. Okay, so it's very surprising to find a 9-mer appearing 6 or more times with its reverse complement within 500 nucleotides. So now here is I would stop the lecture today, and I will give you the homework problem, which is essentially what is the probability of finding a specific 9-mer six times in a random 500 nucleotide sequence. And consequently, if you can solve the above problem, what is the probability of finding a specific k-mer m times in a n-mer nucleotide sequence? So this is a combinatorial, I think, combinatorics problem, something you can solve with the tools that you were given in your school. Yeah, I will stop here and take any questions that you might be having. Any questions?
No, sir. Sir, in the last class, we talked about like uh, sequences at -10 and -35. So I told that like